Thanks very much. It's good to see everybody here today. Lovely spring day. I didn't know when I got up that it would be this nice today. It's really great. The job that I set out to do today was talk to you about primordial plants, the earliest flowering plants, but also to get you involved in making some observations about some flowering plants and give you some skills that you can use to think about evolution, evolutionary biology, and how to build up a structure for understanding a lot about the diversity of life. These are all angiosperms, various kinds producing beautiful flowers, inflorescences from members of the aster family, large trees, small herbaceous plants, beautiful flowers. And our goal is to figure out something about this diversity. This is one very, very well-known flowering plant. In fact, this plant, it's called Arabidopsis, is the first plant whose genome was completely mapped and sequenced. We think we know every gene in Arabidopsis, and we know the function of a great many of the genes in Arabidopsis. This little plant is a weed in the mustard family, also known as the Brassicaceae or the Cruciferae. And if we focus our examination on just looking at the flower of this little mustard, there are four main kinds of structures. There are, moving from the outside, there are sepals, petals, stamens, and in here where you can't see, there are carpels. Carpels produce ovules which get pollinated, gets fertilized by the sperm cell that's in the pollen grain, and that then grows to be a seed that's encased in a fruit. Here's another way of looking at it. On the outside, the outermost whorl, we call it, is sepals, then petals, stamens, and a carpel. These four basic flower parts, these organs, are always in this order, always from outside to inside. And in fact, the molecular geneticists have helped us understand where they came from in a very deep developmental sense. They've identified several genes, 3 in fact, which if you disrupt them is a direct test of Goethe's hypothesis that flower parts really are modified leaves. If we create a mutant of Arabidopsis that knocks out an A gene, a B gene, and a C gene, we end up with all of the flower parts transformed to leaves. So how do these transformed leaves normally appear? What are flowers normally like? I think the best thing we can do is develop some skills at looking at some flowers and see if we can identify the sepals, petals, stamens, and carpels and see what some of the common flower types are. You can take a flower off, and that'll be your flower to take apart. It's okay if you have to. There are plenty. Everyone have a flower? And the best way to do this is to start from the outside of the flower. Sepals are often, but not always, differentiated from petals. Petals are usually more colorful. Sepals are often just green or nondescript. And the question is, how many sepals do you see on your flower? 4 sepals on the forsythia flower. That's a common number. A lot of plants, a lot of flowering plants, especially dicots, have 4 sepals. How about petals? 4? Okay, another common number. Do you see anything about the petals that's interesting? Do you have one with 5? Can anyone see the stamens? They're pretty small. How many do you have? They're yellow. They're yellow and they're pretty big. 2. 4, 4, 2 begins to define the family of flowering plants that we're looking at. And then inside, you probably can't see too much about what's inside, but on the inside there's one carpel, one ovary. And if you would cut it in half with a razor blade and look at it with a little a lens, you could see it had 2 compartments to it. So it's an ovary with 2 carpels fused together. Anybody know what family this is? The name of the plant is Forsythia, and plants are grouped together into families that help us basically predict their characteristics and understand some of the variation. It's in the olive family, or the Oleaceae. So is the ash tree and some other common plants around this area. But olives are in the Oleaceae, as is Forsythia.